My dear students, Assalamu alaikum. I hope you all are fine by the mercy of Almighty Allah. May Allah forgive and bless all of us in the month of Holy Ramadan. Our third card is already started. In this card, I will teach you renal biochemistry. Today I shall deliver the introductory lecture on renal biochemistry. Renal system involves two kidneys, two ureters, one urinary bladder and one urethra. Kidneys are the primary excretory organ of the body. Histologically, kidney is composed of outer cortex and inner medulla. Nephron is the structural and functional unit of kidney. There are about 1.5 millions of nephrons present in each kidney. So, functions of an individual nephron are collectively represented as the functions of the kidney. So if we read one nephron, if we read or know the functions of one nephron, then we can know the functions of whole of the kidney. Now come about the parts of nephron. There are two parts of nephron grossly. One is the glomerulus, another is the renal tubule. Glomerulus is a tuft of glomerular capillaries arising out afferent arteriole and again converting to efferent arteriole, and it is present, glomerulus is present in the cortex, that is the outer portion of the kidney. Here you have to understand the things is this, that when the renal artery enters into the kidney at the place of renal pelvis or hilum of the kidney, it is branched in many branches, and ultimately the arterioles comes out, and these arterioles enter into the glomerulus in the form of capillaries, and that's why the glomerulus is termed as tuft of capillaries, consisting of tuft of capillaries. These capillaries from the artery comes, that artery is, the branch of that artery is termed as afferent artery. The term is afferent artery. You have to remember this. Afferent artery enters the glomerulus, and after the capillaries, uh, there is the filtration, which we will read afterwards. There is the then under high hydrostatic pressure, there is filtrate, and again these capillaries are out comes out of the glomerulus in the name efferent arteriole. So you have to keep in mind that afferent arteriole enters into the glomerulus and efferent arteriole comes out of the glomerulus, and this glomerulus is present in the cortex, the outer portion of the kidney. This is very vital. You have to understand this, that though the artery passes in the hilum of the kidney or the pelvis, it passes through the uh, full portion of the kidney and goes at the cortical region, that the outer portion of the kidney where the glomerulus lies, and there after the smallest branch of the artery, that is the arteriole, enters into the glomerulus and there forms the tuft of capillaries in the glomerulus, which ultimately again comes out in the name of efferent arteriole, and which will go again into the blood. So, and it lies in the uh, cortex of the kidney. The glomerulus lies in the cortex of the kidney. This portion you should be very clear, having the concept that the glomerulus lies in the cortex of the kidney. Then it is the glomerulus is enclosed within a capsule known as the Bowman's capsule. This Bowman's capsule is composed of uh, epithelial cells, and the total glomerulus is encased in Bowman's capsule, which is cup-shaped, and this within the cup-shaped, the glomerulus is present, and it is occupied in the Bowman's capsule. This is composed of epithelial cells. The other term is the glomerular capillaries are covered by epithelial cells, and the total glomerulus is encased in Bowman's capsule. So these are the two things. One is the glomerulus, and its capsule and it is encased in the Bowman's capsule, and the blood supply in the tuft of capillaries. Efferent artery enters, efferent artery comes out. These things you should be clear in conception. Now comes the renal tubule. It consists of proximal tubule and distal descending loop of Henle, then the ascending loop of Henle, and the distal convoluted tubule and the collecting tubule. First, the proximal tubule. It consists of proximal convoluted tubule, descending loop of Henle, ascending loop of Henle, and distal convoluted tubule and the collecting duct. So, what are the parts of renal tubule? The renal tubule consists of proximal convoluted tubule, descending loop of Henle, ascending loop of Henle, distal convoluted tubule, and lastly collecting duct. So these are the parts of renal tubule. Fluid that entered into the glomerulus now is filtered from the glomerular capillaries, flows into the Bowman's capsule, then into the proximal tubule, which lies in the uh, cortex of the kidney. From the proximal tubule, fluid flows into the loop of Henle, which dips in the renal medulla. Now it comes to the renal medulla. Each loop consists of a descending limb and an ascending limb. The walls of the descending limb and lower end of the ascending limb are very thin and termed as thin segment of the loop of Henle. It is to be clear, this is very important because there is a reabsorption takes place. There is the epithelial lining is very thin. That's why this descending limb and the lower end of the ascending limb, the other name is thin segment of loop of Henle. After the ascending limb of loop has returned partway back to the cortex, its wall become much thicker and is referred to as thick segment of the ascending limb. So one is thin segment, another is the thick segment. At the end of the thick ascending limb in a short segment known as the macula densa. Beyond the macula densa, fluid enters into the distal tubule lies in the renal cortex. Now it's again passes to the renal cortex from the renal tubule medulla. This is followed by connecting tubule and the cortical collecting tubule, which lead to the cortical collecting duct. 8 to 10 cortical collecting ducts join to form a collecting duct. 8 to 10 cortical collecting ducts join to form a collecting duct that runs downward into the medulla, again into the medulla, and becomes medullary collecting duct. This forms the medullary collecting duct. The collecting ducts merge to form progressively larger ducts that empty into the renal pelvis. Each kidney there are about 250 large collecting ducts, each of which collects urine from about 400 nephrons. As the glomerular filtrate flows through the tubules, over 99% of its water bearing amounts of its solutes are normally reabsorbed. As I mentioned in the thin segment, there is occurs the reabsorption also into the vascular system, and small amounts of some subs are also secreted into the tubules. There is reabsorption and simultaneously there is secretion also into the tubules. The remaining tubular water and dissolved substance become the urine. So these are the about all are the structures of the kidney in very short description. Now, as you have said, one is portion of nephron lies in the cortex, another portion lies in the medulla. On that basis, nephrons are of two types. One is cortical nephrons, another is juxtamedullary nephrons. In number, cortical nephron are about 80 to 85% of total number, and juxtamedullary nephron is 15 to 20% in number. Cortical nephron lies in the outer cortex and the juxtamedullary lies in the inner cortex and near the medulla. Loop of Henle of cortical nephron is short and the loop of Henle of juxtamedullary nephron is long. Extension of loop of Henle in respect to cortical nephron not beyond the outer medulla, but in case of juxtamedullary nephron it dips into the medulla. In respect to vasa recta, it is absent in cortical nephron and it is present in medullary nephron. Cortical nephron is not involved in the counter current mechanism, but the juxtamedullary nephron is involved in the counter current mechanism. Now comes to the functions of the kidney. If a single function, the function of kidney is the formation of urine. The very important function of kidney is the formation of urine. Secondly, maintains the internal environment and body fluid osmolarity by regulation of water balance, electrolyte balance, and acid base balance. It maintains the internal environment by maintaining the water balance, electrolyte balance, and acid base balance. It's very important. Thirdly, excretion of metabolic waste products. Fourthly, it conserves the nutrients and other valuable substances. It regulates the blood pressure and it synthesizes hormones like renin, erythropoietin, 125 dihydroxycholecalciferol, as you have read in vitamin D, prostaglandin, endothelial derived releasing factor, that is the in short EDRF, and finally metabolic functions, gluconeogenesis and deamination. In this discussion, in this lecture, the important thing is you have to understand the basic uh, concept about the kidney in relation to its structure, a little bit in respect to physiology, and finally the functions of the kidney. There is the formation of urine, maintenance of internal environment, there is the excretion of waste products, conservation of nutrients and other valuable substances, maintains the blood pressure, synthesize hormones, and metabolic functions like gluconeogenesis and deamination. This is very important for your oral examination. Many of the examiners in professional examination asked about the functions of the kidney, so you have to um, uh, remember this um, about the function of the kidney so that you can easily answer about the functions of the kidney. So in the next class we shall go in real sense of renal biochemistry like GFR, which is very important. Today this much. Thank you all. Allah Hafiz. Be fine.